Yeah, sure. I mean, it's just send a link, or I think if we send the link again, it might work because the one that got sent around, you can't just click on on the website or the Zoom call site. It's got to be copy and pasted, so people are probably having trouble signing in. Whereas usually it lights up blue, and you just click on it and you're in. So I don't know what the difference is. You know how when you send the Zoom website, it normally shows up as something you can click on it. Well, this time it didn't, and I think that might be the reason why people are having trouble signing in.
Okay, I will send it again. So, Doctor, do you have Dr. Nathan? Are you ready?
I'm ready when you are.
So,
yep.
Yeah, I think it's better to send it. Gosh, I don't know why sometimes I'm looking at all the previous ones. Why it doesn't come through? Maybe if you send it to another one of the groups. I see the 22-person group. There's another group I have listed as Nitro Medicine. And
Okay.
that one has checking to see.
Yeah, you have the link, right? Can you share the link?
Me?
I'm sharing it right now. Again,
Yeah,
did you see it?
I've got the. Yeah, I've got it under Nitro Medicine Academy, and
Right.
it's got 15 people on it. I'll send it to you. I'm just gonna.
Okay, I did send it. I did send it to Nitro Medicine as well.
Okay.
Let me see.
That has like 15 people on it.
Yeah.
Okay, let me see if that lights up the right way. You know what I'm saying? Like it's got to be easy to click on and.
Right.
No, you
yeah,
sent.
yeah.
I think,
I understand what you're saying.
No. You sent it to.
I just sent it right now. Just now. You can see it right now.
Okay, there it is.
And Dr. Klatz.
Oh yeah. Yeah, that looks like
Dr.
it. Well,
Klatz, can you text me the phone number again? Because I lost it. Adding people and then doing the recording thing. Can you just please text it to me? And see, let me see the recording here. Meeting setting.
I'm recording this on my computer too.
Okay, I'm going to record in the cloud.
Yep.
As well. Okay, so go ahead, Dr. Nathan. If you can introduce yourself and move forward while I get people on the board.
No worries. Well, thanks for allowing me to give an update. I look back and I think in September of 2020, I presented last on our nitric oxide kind of drug discovery pathway and a number of products we've had on the market over the past 15 years. But just by means of introductions, my name is Nathan Bryan. I've been in academic medicine now for more than 20 years, and really my research program has been designed to answer 4 fundamental questions. How does the body produce nitric oxide? What goes wrong in people that can't make it? What are the clinical symptoms or consequences? And then how do you fix or restore nitric oxide production? So I have several dozen issued patents, published hundreds of peer-reviewed papers, and I receive royalties on these patents from the University of Texas. So those are my disclosures. We formed a number of companies, including most recently Nitric Oxide Innovations, where we have some oral drug and topical drugs in the phase 3 FDA approved trials for COVID. We're moving into ischemic non-obstructive coronary artery disease. We've got an IND for ischemic heart disease, and also a topical nitric oxide for diabetic and pressure ulcers. So there's my disclosures. So what I thought we would do is just really kind of review the science and where we're taking the field in terms of new drug development, which for me, trained as a drug discovery chemist, that's what our job is, not really to make dietary supplements or nutritional products, which we've been successful at, but really developing safe and effective nitric oxide-based therapies. And as a basic scientist, my job is to see what everyone else has seen, but to think what no one else has thought, and to give you clinicians the tools you need to treat, prevent, and cure or diagnose disease. So in the broader things, I like this concept here because in my 25 years in academia and science, All disease is caused by two things and two things only. The body's missing something that it needs, or it's exposed to something that it doesn't. And it doesn't matter if this is diabetes, vascular dementia, Alzheimer's, cardiovascular disease, erectile dysfunction. The end result of that is inflammation, oxidative stress, and immune dysfunction that shuts down nitric oxide production, and chronic disease sets in. So the way you can look at this kind of non-invasively is through some new imaging modalities, whether it's SPECT scans or angiography. But the work of Daniel Lehman, who most of you are familiar with, looks at SPECT scans, which is really how well perfused an organ is. In this case, on the brain, what you're looking for is nice contoured imaging here on SPECT, meaning that every region of the brain is getting adequately perfused, has adequate circulation. We're an unhealthy brain. This is I believe this is an image from a former football player with post-concussion syndrome and developed an addictive disorder. And you can see the holes in the brain here are reflective of decreased blood flow or ischemia and hypoxia. So it doesn't matter if it's the brain or looking at angiography in the coronary arteries. If there's a disruption in the blood supply to that organ will fail. And it can be an acute MI here, such as in the left circumflex. And if you don't restore the blood flow in a timely manner, that person will die. But you can see here, if you aspirate the embolus here, you can restore normal function when you adequately reperfuse myocardium. So the whole point of this, whether it's the brain and vascular dementia or acute MI or obstructive coronary disease, the problem is insufficient blood flow. So this is kind of the take-home slide, if you don't remember anything else, that nitric oxide deficiency causes chronic disease, increases sickness and mortality from viral infection, including coronavirus. So the most important thing you can do is enhance your body's ability to make nitric oxide. So this is kind of a restatement of that. So the holy grail in really any management of any age-related chronic disease is restoration of blood flow and perfusion, so that the individual cells can get the oxygen and nutrients they need, and then the metabolic waste products can be disposed of. The fundamental question is, if we're going to restore the function of all organs, tissues, and cells in the body, then how do we restore blood flow and perfusion to these tissues? And that's the role of nitric oxide. It's recognized as a vasodilator. It's a free radical, meaning it has an unpaired electron in the outer orbital. But this nitric oxide molecule is an electron donor. So most free radicals, including oxygen radicals, scavenge electrons from other molecules, and they're oxides. Where nitric oxide donates this electron because there's a polarity between this N and O bond here, and nitric oxide likes to release this electron. So any electron donating species is anti-inflammatory, antioxidant in nature, controls the potential hydrogen or pH of the cell, and affects that you're releasing an electron here. And this is a famous quote by Val Fuster, that the discovery of nitric oxide and its function is one of the most important in the history of cardiovascular medicine. This is a very important study here that's really come to, its importance has been realized in the last couple of years with COVID because of the loss of oxygen saturation in COVID patients. And even when you put them on a mechanical vent or 100% oxygen, many times you can't improve their oxygen saturation. And that's explained by this study here published in 2015. Nitric oxide is required to be bound to a single cysteine residue, the beta-cysteine 93 residue on hemoglobin. When nitric oxide is bound, when the blood goes from the arterial to the venous transit in the capillaries, and hemoglobin undergoes this R-to-T transition, allosteric transition to release oxygen, pick up CO2. If nitric oxide is not bound to that cysteine residue, then oxygen will not come off and you disrupt the cardiorespiratory cycle. So this diagram here reveals that the cardiopulmonary cycle is a three-gas system. It's oxygen, carbon dioxide, and nitric oxide. You cannot deliver oxygen or take up oxygen without nitric oxide bound to hemoglobin. So you have to restore the functionality of nitric oxide in these patients in order to improve oxygen saturation. And I'll show you data from our COVID trial. We're seeing patients go from an oxygen saturation of 78 to 96 within 5 minutes of taking our nitric oxide drug. And that's just breathing room air. That's not even on supplemental oxygen. But it's far more important than that. It's involved in every biological function, from maintaining the integrity of the cardiovascular system, maintaining gut motility as the peripheral nervous system. It's the requisite molecule for stem cell mobilization and differentiation. So it's critically important in regenerative medicine, neurotransmitter in the central nervous system, controls cell cycle. It's a bronchodilator as well as a vasodilator. Most importantly, I think in many conditions is, for erectile dysfunction, when you can't dilate the blood vessels of the sex organs to get engorgement, you develop erectile dysfunction. Those are now symptoms known to be nitric oxide deficiency. And then immunology. So our immune system, our innate immune system generates nitric oxide when we have a viral infection or bacterial infection. We mobilize our immune cells. They go to the site of infection. They generate a lot of nitric oxide through iNOS that binds to the iron sulfur centers of bacteria, shuts down their respiration, and prevents virus from replicating. So in patients who are nitric oxide deficient, they get exposed to a virus. They don't elicit an immune response. They can't mobilize their cardiovascular system or their circulatory system. And the virus takes hold, replicates, propagates throughout the body. And these are the people that are getting sick from COVID. If your body can make sufficient nitric oxide, you've got immune function. Most people, if they get COVID, have mild symptoms, or a lot of people, including myself, who have been exposed to COVID but have never been sick from COVID. And in 1998, the Nobel Prize was awarded for the discovery of nitric oxide to Bob Furchgott, Louis Ignarro, and Fred Murad. And it was Fred who recruited me to join the faculty of UT Medical School in Houston back in 2005, where a lot of the discoveries were in the Institute of Molecular Medicine, where we had a drug discovery program. And it was Fred who discovered that drugs like nitroglycerin work because they were metabolized into nitric oxide that dilated the coronary arteries and explained the mechanism of the anti-anginal effects of nitroglycerin through the production of nitric oxide. So if you can't make nitric oxide, what do your patients look like? Well, they develop high blood pressure. 2 out of 3 Americans have an unsafe elevation in blood pressure. That's a major problem. Erectile dysfunction, 50% of men over the age of 40 report some degree of erectile dysfunction. The list goes on and on. Vascular dementia, diabetes. I'll show you data we published in 2009 showing the role of nitric oxide in insulin signaling. Small vessel disease, that's where we're seeing really great results in ischemic, non-obstructive coronary artery disease, atherosclerosis, blood clotting disorders, including long-haul COVID, platelets become hyperactive, acute respiratory distress syndrome, heart failure. And so these are the exact risk factors for COVID, or really the seasonal flu. So if you have these underlying comorbidities and you get exposed to a pathogen, you're not going to do very well. So if we go back to the basics of vascular biology, that a man is as old as his arteries, we know that the older we get, a lot of this is dependent upon diet and lifestyle, but over time you start to get plaque deposition under the lining of the blood vessel, and this inflammation, oxidative stress, and immune dysfunction leads to cardiovascular disease. Over time, if this plaque becomes unstable, it ruptures, and that, folks, is the #1 killer of men and women worldwide. So we begin to interrogate, well, where does nitric oxide play a role in this whole process? And what we discovered was that if you have good endothelial function, meaning that your endothelial cells can make sufficient nitric oxide, then it prevents the inflammation, oxidative stress, and immune dysfunction, and it prevents the onset and progression of cardiovascular disease. But when you lose the ability to make nitric oxide and develop endothelial dysfunction, then that functional change precedes these structural changes by many years, sometimes decades. So the holy grail is to maintain normal endothelial function and nitric oxide production. So about 25 years ago, we asked a very fundamental question, and unfortunately, nothing's changed. Despite billions of dollars being spent on cardiovascular disease, it's still the #1 killer of men and women worldwide. And this was kind of a headliner here. The amount of people dying from cardiovascular disease is equivalent to 4 jets crashing and killing everyone on board every hour, every day, each year. And to me, that's unacceptable. We know what causes cardiovascular disease. It's lack of nitric oxide production. We certainly know how to diagnose it with the number of functional tests. And now we know how to cure it and treat it. It's through nitric oxide. So it's not that the science is lacking, it's that the education and awareness around from cardiologists and primary care docs don't have the knowledge or awareness of nitric oxide or its potential as a therapy. So in order to treat something, you got to know what causes it. So what causes cardiovascular disease? Well, it's not cholesterol. I think that's probably the biggest myth ever perpetuated in the medical literature. Smoking certainly does. Poor diet, poor oral hygiene, oral infection, sedentary lifestyle, inflammation, even some drug therapy. So what's the common denominator in these seemingly unrelated risk factors? Well, all of these lead to a disruption in nitric oxide production. So let's get started in diabetes. We all know the numbers here. It's a worldwide epidemic. As more countries become westernized, the incidence and prevalence of diabetes shoots through the roof. And we've known for many, many years that people who have diabetes through advanced glycosylation end products, the hyperglycemia, hyperinsulinemia, all lead to a loss of nitric oxide production. And this leads to increased cardiovascular disease severity. So people don't die from diabetes typically unless they go into ketogenic shock. They die from the vascular complications of diabetes. If you knock out the enzyme that makes nitric oxide in mice, their first phenotype is insulin-resistant type 2 diabetes. So this told us if you become deficient in nitric oxide, this through some unknown mechanism at the time leads to insulin resistance. If not corrected, leads to diabetes. And then once you get diabetes, this further suppresses nitric oxide production. And that is the reason diabetics are poorly managed today, because you're not intervening in improving nitric oxide and insulin signal. So I'll show you the data on that. So if you take these eNOS knockout mice, they become diabetic, and the only thing we do is put them on inorganic nitrate. So this would be the equivalent of eating 150 grams of spinach for a normal 80 kilogram adult. So we just give this in the drinking water, these eNOS deficient mice. And you can see here that even though they're eating the same amount of food, drinking the same amount of water, they gain less weight, about a 25% reduction in triglycerides, about a 50% reduction in visceral fat deposition. You can see here that quantified and even visually here. Glucose tolerance improves. Fasting glucose reduces by about 50% or about 30%. Hemoglobin A1C is reduced by 50%, and the ratio of proinsulin to insulin is about 40%. And then their blood pressure is better normalized. So simply by giving inorganic nitrate in the diet, we could overcome the metabolic phenotype in insulin-resistant type 2 diabetes. So this was an interesting observation, but it didn't give us any insight into mechanism. So in 2014, we published this paper where we solved the riddle. So this is the insulin signaling pathway. When insulin binds to the insulin receptor, it initiates a number of protein kinases. These converge on whether it's AMP kinase or PI3-AKT kinase. Both of these enzymes or kinases phosphorylate two serine residues, which is supposed to turn on the eNOS enzyme. But we know in diabetics that this enzyme is uncoupled, not functional. So it doesn't make nitric oxide. So then what happens? Well, this is where the literature led us astray, because we didn't know anything after nitric oxide. But the terminal step in glucose uptake is GLUT4 translocation from the cytosol to the membrane, where it binds glucose and internalizes it. So we got some green fluorescent protein, and we tagged this to the GLUT4 transporter. And you can see here, this was, again, here's the reference here for our paper. But when we activate the GLUT4, either by giving an NO donor or just inorganic nitrite, you can see that the periphery of these cells start to light up. So we can visualize in real time GLUT4 translocation from the cytosol to the membrane, increasing glucose uptake. And this was our model here. So there's two cysteine residues that are conserved throughout evolution in this GLUT4 protein. And post-translational modification by NO, nitrite, or S-nitrosoglutathione is necessary and sufficient for GLUT4 translocation to bind glucose and internalize it. So if you restore the functionality of nitric oxide, you potentiate insulin signaling, and all of this can be explained by the better blood pressure management, the increase in glucose uptake, the decrease in insulin secretion, and the amelioration of the hyperinsulinemia and hyperglycemia.
Can I just interrupt a little bit here, Dr. Nathan, just to confirm what you're saying? Metformin is nitric oxide donor.
No, metformin is an AMP kinase activator, but AMP kinase activates eNOS, but if eNOS is uncoupled, you don't complete this insulin signaling pathway. So the whole
but
point
it does
is,
increase the nitric oxide donor. That doesn't. It does increase. It does increase nitric oxide bioavailability.
no, it does not. You have to recouple this
sure?
enzyme here. Absolutely. There's nothing on the metformin or glucophage molecule that would render it a nitric oxide donor. or an activator. Under normal circumstances, if a patient has good endothelial function and the eNOS is coupled, you can actually increase nitric oxide production through metformin because you can increase the phosphorylation and turn this enzyme on. But if you have an uncoupled eNOS and ED or endothelial dysfunction, then the phosphorylation of these serine residues does not turn the enzyme on because it's uncoupled.
Okay.
And so this here, your ability to make nitric oxide predicts your exercise capacity. So this is in well-trained athletes. The improvement in the increase in plasma levels of nitrite predict your exercise capacity. If you do the same experiment in older patients on an exercise stress test, their inability to increase nitric oxide production, to perfuse the myocardium and dilate the coronary arteries, they fail on exercise stress test. So indirectly, an exercise stress test is looking at the ability of your coronary arteries to make nitric oxide, because there's already 100% consumption or extraction of oxygen. So the only way to increase oxygen availability is through vasodilation. The only way to increase vasodilation, coronary arteries, naturally is through nitric oxide, or you can use adenosine or some endothelium independent vasodilator. But this is the inability to produce nitric oxide upon increased metabolic demand on the heart. So the fundamental question is, how do you control and regulate nitric oxide production? If we can figure that out, then you can cure a lot of chronic age-related diseases. And in 50 minutes, I don't have time to go into a lot of the biochemistry, but there's two ways the body makes nitric oxide. One is through this enzyme nitric oxide synthase, 5-electron oxidation of arginine. The other is reductive pathway. You take inorganic nitrate, convert it to nitrite and nitric oxide. So one can compensate for the other, but if you can't make nitric oxide from either of these pathways, then bad things happen. And here's the enzymology here. So for years, people thought, well, I'll just give L-arginine and hope that you get nitric oxide out at the other end. Well, that's probably the most naive and sometimes dangerous approach you can take, because if you have an uncoupled NOS and you give extra or supplemental L-arginine, then this enzyme generates superoxide instead of nitric oxide. So this uncoupled NOS, there's a disruption in the flow of electrons. So when it's coupled, you take this guanidino-nitrogen here, and through a 5-electron oxidation, generates nitric oxide gas, and you get L-citrulline as a byproduct. But if it's uncoupled, you actually generate superoxide instead of nitric oxide. The other thing, which isn't captured on here, is if you give a lot of L-arginine, you increase the expression of arginase, and you divert the arginine away from NOS and through urea disposal and through arginase, ornithine production, and urea. So this is how it's designed to work. This would be the single layer of endothelial cells. This is the smooth muscle that surrounds all blood vessels. When we begin to exercise, this causes shear stress. That's why exercise is medicine, because it activates the nitric oxide synthase enzyme. If you have good endothelial function, this enzyme makes nitric oxide. And then part of that diffuses into the smooth muscle, where it activates an enzyme called guanylyl cyclase. Guanylyl cyclase converts GTP into cyclic GMP. And cyclic GMP is the second messenger that leads to the calcium-dependent smooth muscle relaxation. The other thing I want to clear up here is Viagra is not a nitric oxide donor. PD5 inhibitors do not affect nitric oxide production. They potentiate nitric oxide-based signaling. So when nitric oxide is produced, it leads to an accumulation of cyclic GMP. These PD5 inhibitors prevent the breakdown of cyclic GMP. That's why you're warned against 4-hour erections and unsafe drop in blood pressure, because now you've lost regulation of the off switch. But we know that 50% of the men that are prescribed Viagra don't respond with better erections or better symptoms of BPI or BPH. And that's because in these patients that don't respond, they're not making enough nitric oxide to lead to any appreciable accumulation of cyclic GMP. So erectile dysfunction, again, is a symptom of insufficient nitric oxide production. It's not a symptom of an overactive phosphodiesterase enzyme. So that's a very important concept, because I hear people all the time, well, Viagra, Cialis, and these are nitric oxide donors. That's what they do. They're not nitric oxide donors. They potentiate and they're dependent upon nitric oxide production. If we look at endothelial function with age, we lose about 10 to 12% of our endothelial function per decade. By the time we're 40 years old, we only have about 50% of the nitric oxide we had when we were younger. So what about the other pathway where we convert nitrate that's found in our food through the nitrogen cycle? So when the air we breathe is 78% nitrogen. This nitrogen is fixed primarily through lightning or nitrogen fixing bacteria in the soil to form nitrate, nitrite, ammonium. Then any plants or vegetables that are grown in the soil, we're getting a source of these nitrogen oxides. And so this is called nitrification. The human body has a denitrification. So it takes inorganic nitrate that's found primarily in green leafy vegetables. 90 minutes after you consume nitrate, it's taken up in the duodenum, the proximal gut, and then our body concentrates it in our salivary glands. So usually about 60 to 70 minutes after you consume a vegetarian plant-based diet, you start secreting nitrate in your salivary glands. And then there are bacteria, nitrate-reducing bacteria that live on the dorsal part of the tongue that reduce nitrate to nitrite. And humans do not have this enzyme. So without the bacteria, nitrate is inert in humans. And then now our saliva becomes enriched in nitrite from the 2-electron reduction of nitrate by the bacteria. Then we swallow our own saliva, which is enriched in nitrite. And when it hits the acid environment of the stomach, it becomes protonated. The pKa of nitrite is 3.4. When it becomes protonated, it generates nitric oxide gas in the lumen of the stomach. And that NO that's formed from swallowing our own saliva in the acid environment of the stomach kills H. pylori, will kill any foodborne pathogen from E. coli, salmonella, botulism toxin. It enhances gastric mucosal blood flow to enhance nutrient absorption. It inhibits gastric ulceration through chronic NSAID use. It's a fundamental physiological process that our body needs to make nitric oxide. And I'll show you how most Americans are disrupting this. But first, let's answer the fundamental question of how much nitrate do we need? If you review the literature, you'll see that you need 300 to 400 milligrams as a bolus. We sampled the standard American diet, what we call the SAD diet, and we're only getting about 150 milligrams of nitrate per day. So just as most Americans are deficient in magnesium and vitamin D and iodine and selenium and chromium, we become a nitrate deficient population. We sampled different dietary patterns, and this dotted line here is the threshold of nitrate that you would need to lower your blood pressure. So the Japanese diet far exceeds this. The dietary approaches to stop hypertension, which is clinically effective at reducing blood pressure, all contain sufficient nitrate to lower your blood pressure. All these other dietary patterns fall well below what's needed to generate nitric oxide. Then the question was, well, how much broccoli or celery or spinach would I need to eat to get 300 or 400 milligrams of nitrate? And we published this in 2015, and we went to Chicago, Dallas, Los Angeles, New York, and Raleigh. We bought food off the shelf from Whole Foods. We brought it back to the lab, ground it up, extracted the nitrate, and measured it. And there's a lot of numbers on here, but let me just make this very simple. So if you lived in Dallas or Los Angeles, you could go and buy 5 stalks of celery, 5 to 6 stalks of celery, eat that, and get enough nitrate to normalize your blood pressure. If you lived in New York, you would have to eat 30 or 40, 50 stalks of celery in order to get the same amount of nitrate from eating 5 to 6 in Dallas. So there are regional differences in the nitrate content of vegetables, and they're different across vegetable categories. And we also compared this to organically grown vegetables. Organically grown vegetables have on average a 10 times lower nitrate than conventionally grown. And to get an organic label, you're not allowed to add nitrogen-based fertilizers to the soil, so the soil becomes nitrate deficient, the plants become nitrate deficient, and without nitrogen, the vegetables cannot assimilate other nutrients. There's data now from 1945 to 2021, and 78% reduction in all trace minerals and nutrients in the food that's grown in the US. So the pressures of feeding a growing population has led to nutrient depleted foods in the form of nitrogen. So the point here is you cannot eat enough organic vegetables to get enough nitrate in your diet to normalize your blood pressure. The other problem is mouthwash. There's 200 million Americans that wake up every morning and use mouthwash. There's 200 million Americans that have an unsafe elevation in blood pressure. It's not coincidental, it's causal. There's 200 million prescriptions written for antacids, specifically PPIs, not counting the over-the-counter purchases that are made every year, and over 200 million prescriptions for antibiotics. So the mouthwash and antibiotics is disrupting the oral microbiome, shutting down the nitrate reduction. The use of antacids shuts down nitric oxide production in the lumen of the stomach. And here are the data. If you take normal intensive patients, this is a human study from 2013, and the only thing you do is give them mouthwash. 7 days later, their blood pressure goes up. Now, this is a very interesting observation, but it didn't tell us what was going on in the management or the regulation of blood pressure. So we started delving into this almost 20 years ago. And this made sense to us because the microbiome provides 3 million gene products. The human genome is only 23,000 genes. So these bacteria are doing things that we as humans can't do or yet haven't evolved to do. They provide essential metabolic byproducts, a true symbiosis. And if we disrupt this, disease ensues. So we started with an IRB at the Texas Medical Center. We did tongue scrapings of kind of patients, faculty, and staff at the medical center. And we just tried to culture these bacteria to see if we could identify these nitrate-reducing bacteria on the tongue. And then did this have any relevance to their blood pressure? And we published this in 2014. The people who had normal blood pressure had the highest diversity in the oral microbiome. Pre-hypertensive patients had less diversity, and people with full-blown hypertension had the least diverse oral microbiome, and they didn't contain any nitrate-reducing bacteria. So again, a nice association, but not causation. Next, we went back to the IRB, and in collaboration with the dental school in the University of Texas Dental School in Houston, we took young, healthy individuals, mainly young dental students, oral hygienic students, medical and dental school faculty, did a full perio exam to make sure there was no periodontal disease, gingivitis. And the only thing we did was we give them chlorhexidine mouthwash twice a day for 7 days. We did daily tongue scrapings and monitored the blood pressure. And here are the data. We published this in 2019. And this is one patient. This was a 21-year-old dental student who was a triathlete in great shape. In 7 days of using mouthwash, we made him clinically hypertensive. In fact, his systolic blood pressure increased 26 millimeters of mercury 7 days after using mouthwash. In 4 days after we stopped the mouthwash, the blood pressure normalized. We were able to identify the bacteria that disappeared when his blood pressure went up, and we identified the bacteria that reappeared at the same time his blood pressure normalized. We sequenced these bacteria. They were expressing these nitrate and nitrite reduction enzymes, so telling us that the presence of these bacteria are important in regulating systemic blood pressure. And this was kind of what we thought was our money slide here. We filed a number of patents on this, but we could predict your steady state blood pressure by the presence and number of these 4 bacterium on the dorsal part of the tongue. This explains a lot. This explains resistant hypertension. So as most of you know, resistant hypertension are patients that put on antihypertensive therapy, but yet their blood pressure is not managed. So this tells us that hypertension is not a renin-angiotensin problem. So ACE inhibitors, ARBs aren't going to affect this. It's not a dysregulation of calcium flux, so calcium channel antagonists aren't going to affect it. It's not a fluid imbalance, so diuretics aren't going to affect this. It's a symptom of oral dysbiosis. So no wonder these drug therapies don't work because, as I mentioned, it's the wrong target. So how do we overcome the variability in the nitrate reduction based on differences in the oral bacteria? Because every one of us has a different oral microbiome, probably different diet, different oral hygienic practices. So again, we go back to the basics. And if we want to prevent cardiovascular disease or end organ dysfunction, we've got to maintain the integrity of the blood vessel. So this inflammation, oxidative stress is when monocytes and T cells start sticking to the lining of the blood vessel. They migrate through, they elicit an immune response, and cause vascular disease. So this is real-time intravital microscopy. We published this in 2009. If we feed an atherogenic diet, you can induce vascular inflammation. So you can see here, there are a number of immune cells that are stuck to the lining of the blood vessel, a little bit of blood getting through here. There's a number of cells that have already migrated through. So this is a heightened state of vascular inflammation, the earliest stages in the onset and progression of cardiovascular disease. If we take that same atherogenic diet, but we just give either nitrite or nitrate in the drinking water and supplement it and restore nitric oxide production at the level of the endothelium, you can see here, even in the face of an atherogenic diet, the blood vessel is more dilated, these immune cells home along the endothelium, but they don't stick and they don't migrate. So this completely abrogates the inflammatory response to a poor diet simply by restoring nitric oxide production. So now that we were armed with that, we go, okay, now we can intervene. And so again, we ask the question is, if we improve nitric oxide production in the myocardium, we protect the heart from infarct, from a heart attack. And we do this in obviously experimental animals. We have transgenic mice where we overexpress the nitric oxide synthase enzyme in cardiac myocytes. You can see here the transfection was successful. The enzyme is active because we're generating a lot of nitrite and nitrate in the blood and the cardiac tissue. And if you subject these mice to a heart attack, so we physically occlude the LAD with a suture for 1 hour, and then we reperfuse for 24 hours, and then look at the amount of injury to the heart. And these hearts here don't see any injury, and this is from 2006. We looked at other tissue. We found that the plasma levels of these mice were elevated in terms of nitrite, nitrate. We harvested the liver of these mice and had the liver of these mice looked like the heart. And just to make sure we had a clean transfection, we weren't overexpressing the nitric oxide synthase enzyme in the liver. You can see here the Western blot shows there's no overexpression of the enzyme. So then the question was, well, does this have any physiological consequence? And so if we occlude the portal blood supply into the liver for 1 hour and reperfuse for 5 hours, the liver is protected from injury, from ischemia reperfusion. So this was the first demonstration. We published this in the Proceedings of the National Academy of Science in 2008, that nitric oxide acted as a hormone. Nitric oxide produced in one organ had endocrine functions and affected the biological and biochemical function of a distal organ. So the nitric oxide that was produced in the heart traveled to the liver, not as nitric oxide gas, because the gas has a half-life of 1 millisecond, but we found two endocrine mediators. It's inorganic nitrite, and NO binds to the cysteine of glutathione and is transported as S-nitrosoglutathione. So we could recapitulate the effects simply by giving inorganic nitrite or S-nitrosoglutathione. Those were the endocrine mediators. Then could we use a supplemental or a dietary approach to replete these tissues? So we put these mice on a standard American diet, a low nitrate, nitrite chow, and then we gave it back in the drinking water. And you can see here, we titrated this in to where we could basically just replete what was missing through the diet to restore normal tissue biochemistry. And again, we subject these mice to 1 hour, no flow ischemia, 24-hour reperfusion. And the mice that don't have enough nitrate in their diet, 50% of these mice don't survive out to 24 hours. And they have about 40% greater infarct in the heart. So not only is it increasing the severity of the infarct, but increasing mortality. If we give nitrite back in the drinking water, 100% survive out to 24 hours, and there's about a 30% reduction in the amount of infarct in the left ventricle. Now, if we take the eNOS deficient mice, so these are completely devoid of any nitric oxide, and we put them on a low NOx diet, again, same thing, and then we give it back, we can replete these tissue levels. And again, we subject them to ischemia reperfusion. And you can see here, this is on the standard chow, enormous infarct here. Again, high mortality, poor cardiac function post-infarct. These that we give it back in the drinking water has significantly less infarct, and echo of these hearts post-infarct have normal ejection fraction, normal cardiac function. So then the question is, how do you restore nitric oxide production? And I tell people you only have to do two things. Stop doing the things that disrupt it and start doing the things that promote it. So what does that mean? If you're using mouthwash, you have to stop. There's clear evidence now that if you use mouthwash, it disrupts nitric oxide production, causes an increase in blood pressure. Fluoride, you have to get rid of fluoride in your toothpaste. Fluoride's an antiseptic, it's a neurotoxin, and it kills your thyroid function. Stop using antacids. You've got to get people off antacids. Your body cannot and will not heal without stomach acid production. Clear evidence now, people who have been on PPIs for 3 to 5 years have a 40% higher incidence of heart attack and stroke. Very dangerous drugs, never approved to be taken chronically, but yet people have been on these for many years, sometimes decades. Stop eating highly processed, high carbohydrate meal, which leads to N-glycosylation end products, glycates the nitric oxide synthase enzyme, uncouples it, you develop endothelial dysfunction. So what can you do? Throw in some more green leafy vegetables, moderate physical exercise. I like infrared light therapy. Better than that is just sunlight, 20 or 30 minutes of sunlight to get the full exposure to all wavelengths of light. And then when all else fails, we have safe and effective nitric oxide product technology. So I'll take you through real quickly. I developed an orally disintegrating tablet about a decade ago, and these are the clinical trials from that. If you put the lozenge in your mouth, it'll dissolve over about 6 minutes, 5 minutes, and as it's dissolving, it's generating nitric oxide gas. So again, we generate nitric oxide in a single biological compartment, and then the endocrine or hormone functions of that nitric oxide do its job. This is a study by Mark Houston in hypertensive patients. You can see here the blood pressure within 20 minutes, about a 4 to 5 millimeter reduction in both systolic and diastolic. 60 minutes later, a further reduction in blood pressure. 4 hours later, long after that lozenge is gone, we see about a 15% improvement in endothelial function, showing that we've recoupled the NOS enzyme and improved endothelial nitric oxide production by about 15%. Here's imaging of the left common carotid. At baseline, we give them the lozenge. 10 minutes later, you see the internal luminal diameter of about goes from 0.52 to 0.6 centimeters. And that 13% increase in blood vessel diameter causes a 34% increase in blood flow. So you want to look at a SPECT scan before and after this lozenge, it's remarkable. And in fact, we've got a pilot study now. We submitted to the FDA to do an Alzheimer's pilot study by giving the lozenge. It's really the beta amyloid, and all Alzheimer's drugs have failed because it's the wrong target. The beta amyloid plaque and the tau tangles are a consequence of insufficient blood flow, getting the good stuff in, the bad stuff out. So our drug therapy to increase cerebral blood flow, to perfuse these regions of the brain, we think we're seeing already clinically an improvement in perfusion, both through SPECT and functional MRI, in 30 days, an improvement in cognition. Pre-hypertension, this was done in Cedars-Sinai, 30 days on the lozenge, we see a 12 millimeter reduction systolic, 6 millimeter reduction diastolic, no change in heart rate, 6-minute walk test, those on the active walked significantly further, those on the placebo got worse. Some cool imaging here, Raynaud's syndrome, microvascular disease, cold hands, cold feet, 10 minutes after the lozenge, we open up the microcirculation, perfuse the digits. Ed Lee, interestingly, did a study several years ago showing that people with already carotid intima media thickness, 6 months later, we're seeing 11% plaque regression. If you compare this to aggressive statin therapy, statins reduce plaque 2.7% over 3 years. We do 11% in 6 months without a drop in cholesterol. One last clinical study here. This was a study I participated with at Texas Children's Hospital, a group of patients with an inborn error in metabolism. So these patients are born without an enzyme called arginosuccinate lyase. They get a buildup of arginosuccinic acid. It's excreted in the urine, and that's their diagnosis of arginosuccinic aciduria. So it's a primary urea cycle issue. So their primary issue is hyperammonemia. But even though they're controlled for the elevation in ammonia, they're still developing liver dysfunction, cirrhosis, blood clotting disorders, resistant hypertension, kidney disease, neurological dysfunction that's independent of the recurrent hyperammonemia. So these investigators at Baylor College of Medicine and Texas Children's approached me to solve this riddle. And we created an ASL knockout mouse. And so we published this in Nature Medicine in 2011. But what we discovered was this ASL protein is what tethers the eNOS enzyme to the membrane. So in these patients that don't have ASL, this protein complex falls apart and they're completely devoid of any nitric oxide production. So this is what allows for arginine uptake into the cell. So the cell doesn't use, the enzyme NOS doesn't use extracellular L-arginine. It uses arginine that comes from citrulline. So there's some intramolecular shuttling of these substrates here. So you get nitric oxide produced, you get citrulline as a byproduct. ASS and ASL convert citrulline to arginine. Arginine is funneled through NOS, get NO. So it's a beautiful cycle here. Without this enzyme, this complex falls apart. And so that explained all the symptoms. In fact, it explained every single symptom, from increase in blood clotting disorder, platelet aggregation, resistant hypertension, and the kidney disease. And this was the lifelong blood pressure of one kid at 5 years of age. He was diagnosed. We published this in 2012, but he was put on standard therapy, and his blood pressure was poorly managed for 15 years. And in February of 2010, he came to Texas Children's Hospital. He was admitted to the pediatric intensive care unit of blood pressure of 210 over 115. Nothing would lower his blood pressure. We knew at that time it was a nitric oxide issue. We started dosing him with isosorbide. 4 days later, he was released from the intensive care unit, sent home, and he did quite well for a period of months, but typical with organic nitrate therapy, he developed tolerance to this medication. And in February of 2011, he came back taking 20 milligrams ISDN 4 times a day, and his blood pressure was 185 over 115. We dosed him with our nitrite lozenge. 4 hours later, his blood pressure completely normalized. He got off all prescription medication, and 12 months out, was very well managed. And so we published this in 2012. His kidney disease resolved in 5 days. He was no longer spilling protein in the urine. His left ventricular hypertrophy and heart disease resolved within 5 months. So normal cardiac function, normal echo data here. And we increased the number of circulating endothelial progenitor cells simply by giving him a source of nitric oxide that his body could not make. So let me move to our topical kind of a program here, because people forget the skin is an organ just like the heart or the brain or the sex organs, and without sufficient blood flow, that organ fails. And so the failing organ of the skin looks like fine lines and wrinkles. You develop dermatitis, acne, poor scar remediation. So we developed a dual chamber nitric oxide serum. When you combine the components of this together, they generate nitric oxide gas. And so these are the published clinical trials here. 30 days, this is using our topical serum. 30 days, you can see these age spots disappear. The inflammation in the chin is pretty much resolved. Inflammation on the neck, this is a 52-year-old lady. 30 days later, you can see the tone, texture, and clarity of the skin improve. The varicosity here, again, clear improvement in the color. The varicose veins go away. This is a lady who had a facelift, and then 2 days later went to her office and started smoking, applying pressure to her chin, and developed a full thickness ulcer. Here we just applied the serum, and I believe along with some exosomes, and within 10 days got complete closure. So no sutures required, no primary closure, just applying the nitric oxide serum with a very good scar remediation. This is polycystic acne. 30 days using the serum, you get complete resolution of the acne. Scarring from teenage acne. This is a 25-year-old scar within 30 days, pretty good remediation here. And nitric oxide is agnostic to skin type or skin color. Works the same in African Americans, Hispanic, Asians, Caucasian. It generates nitric oxide, dilates the microcirculation, improves blood flow. So let me finish up on some of our drug programs here in the next 5 or 10 minutes. Early in June of 2020, we recognized that people that were getting COVID and dying from COVID were those patients who couldn't make nitric oxide. The problem with COVID even back then was loss of oxygen saturation. We knew there was an elevation in D-dimers, so platelets were becoming hyperactive. And then the long COVID symptoms, the vascular inflammation, dysfunction that persisted long after the active infection. And then we knew that nitric oxide actually inhibits the coronavirus replication. And here's the problem, whether it's active COVID or now whether it's the mRNA vaccines giving the production of the spike protein. The spike protein is the toxin. It causes these tangles, the red blood cells to tangle, platelets become activated and causes severe endothelial dysfunction and inflammation. Here's the data from 2005 showing nitric oxide inhibits the replication of SARS-CoV-1. We looked at the health disparities because it was African Americans who were seen to have 10 times higher death rates and hospitalization in the same counties as their Caucasian counterparts. And we knew from years ago that African Americans, the health disparities of African Americans could be explained by insufficient nitric oxide production. And it's primarily due to NADPH oxidase overexpression, G6PD deficiency, all lead to a reduction in nitric oxide. So this made sense to us. So we submitted an IND to the FDA to try to restore and replete nitric oxide production as an early treatment for COVID. And it made sense because if you have low vitamin D, you're going to do very poorly. You get infected with COVID. So what do? Well, you give vitamin D. Same thing. If you're low in zinc, we give zinc. If we're low in nitric oxide, we give our nitric oxide drug. So we submitted the IND of June of 2020. Our IND was approved in July. We got the drug manufactured by Patheon Thermo Fisher in October, and we got our IRB approved in November. We started rolling patients. Our drug is called Noviricid. This is the generic. Nitrovir is our brand name. But we generate about 40 to 50 parts per million NO gas as it's dissolving. We're seeing from our early clinical data, this is a double-blind, placebo-controlled, multi-center clinical trial. We have 26 clinical sites active now in the US. We give people a free pulse oximeter and a blood pressure cuff. So you can see here, this patient probably should have been in the hospital, blood oxygen saturation of 79. We can text them when it gets this low, ask them if they've taken their study medication. They hadn't, they take their study medication. 8 minutes later, we've increased the blood oxygen saturation to 91. A little bit of rebound tachycardia here. The tachycardia resolved in another 5 minutes. So obviously this patient wasn't getting the placebo. So where are we now? We've dosed over 500 patients, high risk, really sick patients with our nitric oxide drug. Not a single adverse event, no unsafe drop in blood pressure, no first dose effect, no headaches, no adverse events of any kind. So now with the FDA, anything we put through goes straight into a phase 3 clinical trial because there's no safety issue and we've cracked the code on the dose. So we've got a drug called nitrocardia we're using for ischemic non-obstructive coronary artery disease, got an oral drug for Alzheimer's that will start in 2023. Ischemic heart disease, about 30 million Americans are living with ischemic heart disease now. The cornerstone therapy is really isosorbide dinitrate. And these drugs have to be weaned on and off because of the tolerance. We found that our NO lozenge does not cause tolerance. In fact, we get the opposite of tolerance. So now we're going into an ischemic heart disease trial, which we think is going to be a game changer for the management of cardiovascular disease and ischemic heart disease. And then our topical, we're taking into diabetic ulcers and pressure ulcers. So here's our dual chamber drug here. This is a plate. I believe this is Pseudomonas. You can see here a very effective antimicrobial. In fact, we see the same with MRSA, strep, staph, any bug that's present on any non-healing ulcer, we kill it. Here's some of our early preliminary data here. Here's a diabetic foot ulcer that was non-healing for a year. We basically heal it in 30 days. Second degree burn here. You can see within 7 days of using the serum, we start to get reepithelialization. Here's a 3-year-old non-healing paraplegic decubitus ulcer that had been seen by the wound care specialist at Yale Medical School in Connecticut. And so for 3 years, this wound would not heal. We started the nitric oxide topical, and you can see the progression here. Within 195 days, we got complete closure. So what was previously non-healing the three previous years, within a matter of half a year, a little over half a year, we healed this wound. So then let me just finish here. If we're developing nitric oxide therapies, is this what nature intended? We don't want to give the body anything that it hasn't seen or what's not intended. So is increasing nitrate in humans what nature intended? We answered this question by this is Lincoln here, who's now 14, but when he was born, we were discharged when my wife gave birth. The discharge nurse, I clearly remember her saying, well, you should breastfeed, but if you don't breastfeed, then give the baby this formula. So when we left the hospital, I took my wife to the lab and we collected some of her colostrum. This was day 2 postpartum. And we compared it to what was in the formula. And what we found in terms of nitrite in the breast milk was extremely high. There was hardly any detectable nitrite in the formula. So then I went and got an IRB and we went around to Memorial Hermann Hospital in Houston and we collected breast milk from new mothers. We divided this into colostrum, transition milk, and mature milk. And we found that the anions, the nitrite-nitrate ratio completely changed. So colostrum seemed to be the highest in nitrite. And this was the data, again, we published this in 2011. But this, we had to think about this. And if you think about this, it makes perfect sense. Babies are born sterile. So they don't have any bacteria to reduce the nitrate to nitrite. So what does nature do? Well, it provides nitrite in the form of the colostrum. Then about day 4 to 5, you start to get bacterial colonization in the baby, and then the breast milk switches to nitrate. Then the baby can then convert it because it has the right communities of bacteria. But we also found this is different in cesarean versus vaginally delivered babies because of the inoculation through the vaginal canal. The other thing, I didn't put it in here, but we basically solved necrotizing enterocolitis. So NEC still has about a 20% mortality in infants. So these are premature babies with underdeveloped lungs. They develop necrotizing enterocolitis, the gut rots. And if we just give nitrite in the formula, we completely prevent necrotizing enterocolitis. The next was on altitude. How do we adapt to low oxygen? Whether it's providing collaterals with patients with chronic ischemic heart disease, they develop collaterals. But even altitude, how do you develop and evolve to adapt to low oxygen from altitude? So we went to Tibet. These people live at about 12,000 feet above sea level, but yet they don't get high altitude pulmonary edema. They walk around, they don't get altitude sickness. So what is their adaptive effect? We do forearm blood flow measurements and find that even though they're breathing in less oxygen because of the decreased partial pressure at altitude, they deliver more oxygen per unit time than we do in the US. We sampled the blood. This is the EPR of their NO bound to hemoglobin. In the US, it's barely detectable. They have a lot of nitric oxide bound to the hemoglobin. They have about 50 to 100 times higher concentration of nitrite and nitrate in the plasma. And so this explains high altitude training. The benefits of this aren't just due to increased erythropoietin and hematocrit formation. And we've done this in Ethiopia now, where we're 6,000 feet above sea level, and they have intermediate levels of nitrite, nitrate in the plasma. So if you're moderately trained or well trained and you go to altitude, your body turns into a nitric oxide generating factor. Then when you come back down to sea level, you're a well-oiled machine that enhances performance. However, if you have poor endothelial function and you can't make nitric oxide to adapt to low oxygen, these are the people who get mountain sickness, high altitude pulmonary edema, and these are the people that do poorly. So let me just wrap up here and reiterate, people get sick for 2 reasons and 2 reasons only. Body's missing something it needs, it's exposed to something it doesn't need. You have to remove the source of exposure of the toxin, replete missing nutrients, give nitric oxide, restore blood flow, and the patient will get better. We're kind of like batteries. Over time, we lose our charge, we lose our nitric oxide, and at some point, we no longer have a charge and we die. It's not like a light switch where you just crash over one day. That's how it should be, to live a full life and then not wake up one day. But we shouldn't live with chronic disease and low charge. So again, follow these rules. Stop using mouthwash, get rid of fluoride, get people off antacids, moderate physical exercise, go in some more green leafy vegetables, get some sunlight, or take some nitric oxide therapy. Be careful. This is a paper I published in PNAS in 2004 where we were trying to figure out where does nitric oxide go and what does it become? There's a lot of companies out there selling nitric oxide products that are doing a lot of harm to a lot of patients because they're forming these nitrosating agents. Peroxynitrite is really a non-issue. This just rearranges to inorganic nitrate, so we don't worry about peroxynitrite formation. This is our simplified model. Here, you got to standardize the amount of nitrate. We've got capsules for that. We've got a nitrite lozenge we're developing as our therapy. Basically gives an exogenous source of nitric oxide with no tolerance development. And we recouple the NOS enzyme. I just conclude that nitric oxide controls and regulates blood flow and oxygen to every cell in the body. Our body cannot and will not heal without nitric oxide. And we see, I think this is going to be the way you treat patients for the next 100 years. There's not a single indication where nitric oxide in the right patient at the right dose at the right time will not make them better. So with that, I'll stop. And here's a couple of references. Those of you interested in a edited read here, a book I edited with Joe Lascalzo, who's chair of medicine at Harvard, Nitrite, Nitrate, and Human Health and Disease. This is my lay book, Functional Nitric Oxide Nutrition. If we don't get questions answered, shoot me an email and I'll be happy to respond. But thanks, Dr. Halassa, for the opportunity to give an update on what we've been doing over the past 2 years.
Quick question.
Yeah.
So So nitric oxide also is involved with oxidative stress, I understand. So is there a point where nitric oxide or excess nitric oxide is a problem, or giving nitric oxide in the presence of endothelial dysfunction? So I'm trying to figure out where the balance is, because my understanding is nitric oxide is also part of an oxidative stress pathway.
Well, it's kind of dose dictates the poison, right? So too little nitric oxide is bad. You're going to get hypoperfusion. Too much nitric oxide is bad because it leads to systemic vasodilation, hypoperfusion, and in-organ injury. But what we do, so again, it's all about the amount. So what we do is we give the body the amount of nitric oxide. So let me step back. There's two measures of toxicity for nitric oxide. One is hypotension. The other is methemoglobinemia. So it'll oxidize the heme of iron and reduce the oxygen carrying capacity of hemoglobin. So in all of our clinical trials, we don't get any methemoglobin formation, and we don't see an unsafe drop in blood pressure. I think what you're referring to is probably this proxynitrite cycle, where you form nitrosative stress and oxidative stress. So one of nitric oxide's critical role is to scavenge. Is to scavenge superoxide. And so NO reacts with superoxide to form this ONOO minus molecule. That's proxynitrite. But this is a cage-like molecule. To stabilize the electrons on this molecule, it just simply rearranges to NO3 minus, which is inorganic nitrate. But if you look at a lot of pathology, histology papers, you'll see nitrotyrosine, and they say, well, it's nitrotyrosine formation, and it's contributing to the pathology of that. But that's not the case. The analogy is, if there's a crime, the cops always show up at the crime, right? So they're there. But that doesn't mean they caused the crime. So just because nitric oxide is around in these different pathologies doesn't mean that it contributed to the pathology. It's there, it's the cops on the scene trying to clean up the scene, to try to prevent the inflammation, the immune dysfunction, and the oxidative stress. So if you have sufficient nitric oxide production, you inhibit NADPH oxidase production of superoxide, you recouple the electron transport chain of the mitochondria, so you decrease superoxide production in the mitochondrium, and you recouple the NOS enzyme, so now you're generating nitric oxide instead of superoxide. So it's a self-fixing mechanism.
Mm-hmm. Dr. Bryan,
how does the peroxy, and by the way, thank you.
Yeah,
go ahead.
No, go ahead.
Hi, James.
My mother, I think we talk and think at the same time or something.
That's no problem.
No, this is me, Dr. Colleen. Along the same lines with Dr. Joseph, when do we have too much pro-oxidative movement? I appreciate the studies you've done, and God knows I want to get some topical NO from my skin. That was a great presentation. However, I find that I don't need NO as much as I used to, but let's not look at it in isolation like your studies are.
Right.
I take metformin, I take methylene blue twice a day right now. Sometimes vitamin C, patients aren't studies. We take a lot of things, and I'm finding less is more. And also, I am allergic to beets, so I do not like getting it in the beet form. But so.
Yep. Well, in our lozenge, we don't have beets in the lozenge. We've simplified this, but you make a good point. And look, my job is, not to sell you product technology. My job is to inform you. But the most important experiment is the N=1 experiment, right? Whether it's an N=1 for me or you or your patient, and everybody's different. I think the problems arise when there's an imbalance in the pro-oxidant versus antioxidant response elements and the mechanism of that. But here's what we know: if you don't make nitric oxide, then you get a lot of superoxide and oxidative stress from NADPH oxidase, from uncoupling of the electron transport chain, and from the NOS enzyme itself. So that leads to a pro-oxidant state just by virtue of not being able to make nitric oxide. But then when you restore nitric oxide, you suppress NADPH oxidase production. You recouple the electron transport chain at complex 1 and complex 3 and 4, and then you prevent the electron leakage from that. So again, it's a self-correcting mechanism, but it's dose dependent. So you can take too much and you can lead to this pro-oxidant environment, because just like this slide here, if you generate a lot of nitric oxide and you still have a lot of superoxide being produced, then you're going to form a lot of these nitrosating agents, because the excess NO, well, you know, this is a second order reaction with oxygen, a second order reaction to form these nitrosating agents, which nitrogen dioxide, which is hugely toxic, N2O3, N2O4, all of these will form nitrosamines and lead to a lot of problems. So again, we have to look at the entire system, but in order to do these studies and gain an understanding, we have to do this in isolation so we can track the biochemistry. But
Do you
again,
have any suggestion about how a person can measure this for themselves and for our patients? Oxidative, you know, where is their oxidative level?
yeah, well, we've that's been a major problem for, you know, that was the question when we first launched my first company in 2010. How do you know if you need nitric oxide?
Right.
And so I developed a salivary test strip that you can look at the amount of nitrite in your saliva. So if your body can convert the nitrate in your saliva to nitrite, we can use that as a proxy on this saliva test strip and give you an indication of if your body's making nitric oxide. But I tell people it's a good tool to have in your toolbox, but it shouldn't be the only tool you're using. So if you have normal blood pressure and you can turn that test strip pink, then you're probably fine. You know, if you eat the right foods and you absorb the right nutrients and you get moderate physical exercise and maintain some good endothelial function, then you're doing fine. But we always want to use diet and lifestyle as a first intervention before we get supplements or even certainly drug therapy. There's no alternative to good diet and good living. But the point is, most people are so screwed up and dysfunctional because they're on antacids, using mouthwash, they're on polypharmacy, and nothing in their body is working. So we've got to do it for them.
Right. Thank you so much. What do you think of Berkeley Life compared to others?
Berkeley's a good company. You know, I consult for them. They have a nitrate capsule. So again, it's, you know, this is really the first one on the market, because we know that, you know, 80% of the people in America are nitrate deficient just because of the foods they eat and where they live and what foods they're eating. So if you got a patient that's low in vitamin D, what do? Well, you give them vitamin D. If you're low in nitrate because they're not getting it in their diet, what do? Well, you give them some nitrate capsules. And then you can actually measure that with the test strips and see that they can improve on the test strip. So it's a single strategy. Again, there's two ways the body makes nitric oxide, and we have to hit them from both ends. So if you're deficient in nitrate supplementation makes sense. But if you're on antacids or on PPIs, then your body's not going to be able to convert that to nitric oxide. So there's limited utility in that. Any other questions?
I have a suggestion.
Okay, I like suggestions.
Yes, with sleep apnea, you develop intermittent hypoxia, which also causes endothelial dysfunction.
That's right.
And the endothelial dysfunction causes nitric acid deficiencies. And you have the same syndrome, really, as you would have with a deficiency from your diet. Could you supplement that, and how would you supplement that? Actually, that's one of the reasons why, even though you can change the symptoms of sleep apnea, you generally do not change the mortality of sleep apnea.
Now, you make a good point. I mean, look, if you have obstructive sleep apnea, you know, you got to fix the obstruction. But the problem is, and you make a very good point, because it's a perpetual cycle. It's a feed forward cycle. You need nitric oxide to uptake and deliver oxygen. You need oxygen to make nitric oxide through the NOS enzyme. So if you have apnea or hypoxia from whatever reason, then you can't make nitric oxide, and then what little oxygen you are getting, you can't deliver it to the cells. So you become systemically hypoxic. And that's the beauty of our lozenge. because the lozenge dissolves in the mouth, it generates nitric oxide. We see an improvement in oxygen saturation, and we see dilation of the carotids within 12 seconds of putting the lozenge in your mouth. So to answer your question, I recommend and suggest that everybody with obstructive sleep apnea or any hypoxia or ischemia take the lozenge, because their body can't make nitric oxide, so they've got to have something that does it for them.
Okay. I also have another question. In elderly people, they have those kind of bleeding under the skin things, thinning skin, frail blood vessels. Would your serum, would that help that?
Yeah, well, I say yes, but look, there's a limit of diffusion of nitric oxide gas in the tissue. So we can't be so naive to think that the nitric oxide applied topically is going to diffuse into the resistance blood vessels and lead to a lowering of systemic blood pressure. So it doesn't diffuse that far. It's a couple of millimeters. So, but these superficial blood vessels, and you can see it because when we apply the serum, we turn the skin pink. So we recruit capillaries and get this hyperemic response. But it's superficial blood vessels. So not certainly arteries and arterioles. It'll dilate the veins and the capillaries, but no deep blood vessels.
But the ones that show on the skin, which
Yes,
really
absolutely.
bother little old ladies, of which I am one.
Right.
Yes. Okay. That's good.
And then I'll go through, I'm looking at the chat here. Is NO okay for active cancer? There's a lot of controversy for NO and cancer because contemporary thinking, and most oncologists will tell you, don't give a pro-angiogenic molecule to a metastatic disease. I tend to argue against that because cancer tumors, active tumors are by design hypoxic. They regulate their own blood supply. They don't like oxygen. It's hypoxic, low pH environments where cancer proliferates and grows. So if you dilate the blood vessels and hit that active the tumor environment with oxygen, then that provides an environment where the cancer can't grow. But again, that's my thought based on the science, but we don't have any clinical data. The other is shown that metformin for type 2 diabetes, metabolizing the intestine subsequently with release. Yeah, not a question, but thanks for those information. Can you get oral effects of NO with the topical? No, typically not. I mean, the good thing about the topical is it's local. It only works the area in which you apply it. Should we take NO with or without food? We recommend taking, a nitric oxide product on an empty stomach. And the reason for that is, especially with the lozenge, so every time you salivate now for the next 6 or 8 hours, you're going to get a burst of nitric oxide in the lumen of the stomach. So if you've got, undigested food particles in there, then those peptides and undigested proteins and cysteine thiols in there will scavenge the nitric oxide before it has a chance to reach the mucosa. What's the best supplement for high blood pressure? I feel like this is a Planet FDA question. So you cannot use a supplement for the treatment of high blood pressure. That's a disease status. So what we do, I mean, you can, the supplements and nutritionals on the markets aren't designed to diagnose, treat, cure, prevent chronic disease or blood pressure. But what we do say is, in our lozenge, the NO2U lozenge, we're seeing very good effects of regulation of blood pressure because we're generating the nitric oxide. Or better yet, most people who have high blood pressure, if they just stop using mouthwash, get rid of fluoride and get off antacids, you'll see an improvement in their blood pressure. So with that, I'm going to respect everybody's time. I think Dr. Halasa said there's somebody coming in here afterwards, but this will be recorded, and I'm happy to upload it on my YouTube channel. And that way people can go back to it. And again, I encourage you to, I've got a website here. It's an educational website. I've got a 6-minute video on nitric oxide. But these are some cool references. I do a monthly blog here. You can follow me on Instagram and YouTube. But my objective is to provide clinicians and patients with information and awareness on nitric oxide to where they can make themselves better or give physicians better tools to make the patients better. So with that, I'll sign off here, and thank you all for your attention.
Well, thank you very much, Dr. Nathan, for your presentation. It was great.
Thank you.
And hopefully we'll see you again.
Absolutely.
Any questions? I'm in Iceland, so it's like 1 or 2 a.m. now. Can you hear me, guys?
Yes, I can hear you.
Yes.
Okay. Right. So, yeah, if there's any question, please, if